It's great to be here. Jacqui, Ishwar, appreciate the introduction. I have a lot of things to talk about, and I'm not sure I'm going to have enough time to go through them. So I may in fact not do as well as Susan did in keeping us on time. So I apologize if that's true. Let's see if I can figure out which one's mine. That is not it, and that's not it either. I guess somebody closed it. To help me figure out where it is. I opened it and I guess somebody closed it. Sorry about that. So some of the things I won't talk about that NCI has been doing are things like the physical sciences of oncology, the work we've been doing in computational biology, and again, there's lots of funding opportunities in these areas, and there are so many different activities at NCI that I'm going to focus really on just a couple. And I apologize for skipping so many really important programs. And let's see if we can figure out where my slides went.
Yeah, that's probably. Does it have your name in it?
Yep.
There we go. Okay.
That's it. Okay, now we're back on track here. So, as Ishwar said, I was asked to talk about the data commons in the age of precision medicine. What I want to do is go through a little bit of background in NCI and the cancer mission that so many of the cancer institutes around the world have. Go through a little bit of why I think open science is so incredibly important, and I want to echo what Susan said about open science really needs to be filled with two-way conversations, and it's not just about getting data open, it's really about what you do with it. We want to talk very briefly about NCI Match, so it's a very important umbrella clinical trial that NCI has been running for the last 2 years. A little bit about proteomics, genomics, imaging, and clinical phenotype, because I think, again, I'll talk a lot about genomics, but I want you all to realize that my view of precision medicine is far broader than just genomics. And then we'll talk about the NCI data commons. So this is something I learned when I was talking with patient advocates that's really important to do, is actually connect at a human level with people. And we often forget about that as scientists, at least I do. And it's been really interesting for me to realize how important it is when you're talking about cancer to recognize the society and the personal impact that cancer has. So, for instance, for myself personally, I lost 3 of my grandparents to cancer. And that's obviously that has impacted me in many different ways, including my professional direction. So I didn't put the long slide of all the different NCI funding opportunities, but I did want to call out 2 in particular. One is ITCR, so that's the Informatics Technologies for Cancer Research Program, and also Cancer Moonshot. So I'll talk a fair amount about the Cancer Moonshot in the next half an hour. And it's important to recognize that we will be dispersing about $1.8 billion US, so that's almost 40 billion of Czech krona in the next 7 years. So again, it's not all going to go to informatics and data science. I wish I could say it was, but a good chunk of it will go to data science. Because again, I think as we all recognize, our ability to do things with data is incredibly important. And in fact, just generating data is not enough. So back to the NCI. So our mission is really to develop the knowledge base that lets us change the impact and burden of cancer in the United States and across the world. And that last year in 2016, there were roughly 1.7 million new cases of cancer in the United States alone, more than 12 million across the world, and 600,000 deaths due to cancer in the United States. and cancer is the second most common cause of death in the United States. But it's not all grim. We've been doing a better and better job of improving diagnoses, and as of the end of last year, again, there were an estimated 15.5 million survivors. So we've been doing a better job turning cancer into a chronic disease, but that itself has some real implications for how we think about patients' health, how we think about improving treatment, so that in fact patients that are living longer have fewer and fewer burdens. And that brings me to the precision medicine definition, and that's that at least I believe that precision medicine is best defined as requiring a fundamental understanding of the complex interplay between genetics, epigenetics, nutrition, environment, the clinical presentation that we see when a patient comes in and gets a diagnosis, and how we then provide the right, effective, evidence-based prevention and treatment for those patients. So I've been very fortunate during the last two plus years to have been very closely connected to the Beau Biden Cancer Moonshot in the United States. And actually I see a number of folks around the room that were part of that as well. And I think what was really exciting about it is one of the three platforms was enhanced data sharing. So that was the first time, for instance, I really heard a prominent US politician talk about things like interoperability and data science. So it was very exciting to hear a level of interest and frankly a focus by then the former Vice President Joe Biden. And of course these things, and I'll bring this up in several different slides, these things require lots of different people to make them happen. And one of the teams that you won't hear a lot about because they were all behind the scenes was the Cancer Moonshot Data and Technology Team, and it was made up of folks throughout the federal government, and they all worked very, very hard with really doing this as part of their nighttime job, not their day job. So they were remarkably effective. And inside the NCI, we were able to then stand up the Blue Ribbon Panel, and the whole idea of that was to create the scientific recommendations necessary for the NCI to define what kinds of programs we need to run to really understand cancer in a more fundamental way. And there are 28 members and 7 different working groups, and the working groups engaged more than 150 people across the country and across the world. So to give you some of those names, here are the folks that were on the core working group team. Tyler Jacks at MIT, Liz Jaffe at Johns Hopkins, and Dina Singer at NCI were the co-chairs. And then lots of folks from all across the country, all across the United States were involved. And the 2 folks in red are patient advocates on the board. I won't go through all the Blue Ribbon Panel working groups, but I do want to call out Angel Pizarro and Gaddy Getz. Angel's here in the audience, and they both ran the working group for the Blue Ribbon Panel on enhanced data sharing. So that was about a year and a half ago now that those working groups were established, and back almost a year ago now, they released a set of recommendations. Those are all available at cancer.gov/BRP. And I'm, again, not going to go through all those recommendations, but the NCI's used those recommendations really as a blueprint for how we come up with funding opportunities for the whole community. And you can see right now, those funding opportunities, some of them have gone through our Board of Scientific Advisors, and they've been recommended, and they will be released for funding opportunities in fiscal year 18. From the enhanced data sharing perspective, really the vision is how do we create a learning healthcare system for cancer? And in my mind, the goal is really how do we learn from the experience of every single cancer patient, not just the cancer patients that go on to clinical trials or participate in clinical research, but how do we really make sure that as a country, as a world, that we learn from the experience of every single cancer patient. And that's got to extend through survivorship and understand the impact of their treatment on survivorship itself. And to make that happen, you know, I guess I'll say I'm a very big proponent of open science, open data, and the need, frankly, for us to work as teams and for us to work in multidisciplinary environments. And again, looking at it from an informatics perspective, I don't think that I'll come up with anyone who argues with that. But it's one of the big reasons I think this is so important is it also accelerates innovation. And the more we can get out into the open, the faster we can change, the faster we can improve. So again, I think that open, the FAIR principles that Susan mentioned earlier, are incredibly important, and I'll talk a little bit more here about FAIR. So Susan already mentioned it's making data findable. I add a couple extra letters. I say it's accessible, attributable, because again, we do need to give credit to individuals that are contributing to different resources, and whether that's data or software or annotations, people do need to get that credit. And we need to be able to understand who's done what with data and what discoveries they make with that. And likewise, all that needs to be interoperable. It needs to be reusable. And again, I think reusability is an incredibly important part of getting to reproducibility, and provide that recognition. And the recognition is incredibly important. So I'm going to switch gears just a little bit. And this has been a really important concept for me, thinking about engaging many of the folks out in the computational community that aren't involved in biomedical research. And that's that cancer itself is a grand challenge. And it requires deep biological understanding. So one of the things that, again, Susan was mentioning with a lot of those different the different science that's been done in the last few years is how little we actually understand about biology, how little we understand how cells migrate, how little we understand about vesicle transport across membranes, and how that actually happens and how critical that is to communication, both in normal cells and then in cancer. And to make that happen, we really do need to have fundamental advances in scientific methods, in instrumentation, in technology, in computation, mathematical models. And I think what's really exciting and complex for all of us is these things are all happening simultaneously, and how do we do a better job of pulling in all that innovation, all that change that's happening? And of course, at the end of the day, what's important for all of us is that really that data that's being generated is critical to creating this learning health system for cancer. Or at least that's my takeaway. The other thing I wanted to talk very briefly about, and I'll bring up one example of this, and then I'll leave it, is that at the end of last year we had a lot of detailed data on roughly 42 cancer types in the Cancer Imaging Archive, in CPTAC, that's our Proteomics archive, and from TCGA, so the Cancer Genome Atlas, from more than 10,000 different patients. And that's been incredibly important and incredibly valuable in elucidating a lot of the underlying biology and genetics in cancer. I think something that's been underappreciated is the value and the power of what's available in the imaging archive. That's not to take away from the other ones, but I think it's gotten a little less attention. And I think that we'll be paying a lot more attention to imaging as we move forward, in part because when we look at things like the SEER registry, where we have population-based data on cancer patients in roughly 30% of the US population, we can start to automate getting imaging from all those patients. So when you think about having access to all those images and what we can do with them, would be a phenomenal resource. So now I'm going to turn to genomics, which is, I think, a more common theme. And something that we've learned is that when you look across different types of cancers, what really matters is the cell of origin and the pathways that have been disrupted. And that, in fact, the same molecular abnormalities that are found in different cancers can arise in different organs. So that was really the origin of NCI Match. It's an umbrella trial. And for those of you who aren't familiar with clinical trials, umbrella trials are where you can run multiple clinical trials essentially under a single approval. And the way that Match was constructed is that each arm is targeted for a set of molecular variants, genomic variants, and then they're targeted with specific agents. And this is a couple slides from Peter Dwyer, the ECOG Akron study co-chair from back the end of May that was given at ASCO. And back in June, the NCI Match trial hit its 6,000th patient in roughly 2 years. That's the fastest accruing clinical trial the NCI has ever run. And that more than 1,100 different sites contributed patients to the trial. So again, it's been tremendous to see how engaged the entire cancer community in the United States has been in Match. What we don't know yet is the outcome of all those trials because it takes some time for the patients to go through and progress or not progress on their therapies. But what's been very interesting is as we've run the different arms to realize what the frequency of some of these variants really are. And almost without fail, the frequency is lower in those populations than we had predicted. So what it's meant is that as we were closing the initial phase of Match is to fill a number of these arms, we need to screen many more patients than we were anticipating. So rather than our initial number of 3,000 patients or 6,000 patients, to fill some of those arms, we need to screen 10,000 or more patients. However, I want to also emphasize some really good parts about Match and realizing that it's not just about matching patients to the best therapy. It's really about making sure that we know what therapies won't work for patients. And that's just as important as knowing what therapy to give. Something else I want to point out is that biology is complex. We still don't know enough about biology to really be able to do as much targeted therapy as we'd like. And that genomics, imaging, and clinical labs are really the first wave of thinking about precision medicine. So as I said at the beginning, genomics is the beginning of precision medicine. It's not the end. Now, I'll probably repeat that multiple times. So now I want to turn to is talking about the NCI data commons, and in particular the genomic data commons, because that really represents a new architecture and a new way of thinking about sharing cancer data. And it's related to the cancer moonshot as well. So back a year ago now, the Genomic Data Commons opens at the University of Chicago. So Bob Grossman is the PI of the Genomic Data Commons, and that's Bob right in the middle. And it's Joe Biden on the left. Let's see if I can get, there we go. Joe Biden and Allison Heath. Thank you, Tony. And Tony's going to be up talking later about the data commons on the right. It was launched at ASCO back on June 6th of last year, and it had roughly 2.6 petabytes of legacy data and about 1.5 petabytes of harmonized data. I also want to point out it wasn't just the University of Chicago that was involved in the GDC. It was also the Ontario Institute for Cancer Research was fundamental to getting the visualization done. and also Leidos Biomed running the contract, making sure that, in fact, everything really did hit all the milestones. And again, something I think is really important, and it ties together with BD2K, is the GDC itself it follows the BD2K initiative and is part of the NIH Commons. So, and of course, we think it's a really important way of making data findable, accessible, attributable, interoperable, reusable, and provide recognition. And by the way, all my slides are on SlideShare. I uploaded them last night, so they're already there. And there's lots of buzzwords down here for folks that love buzzwords. I won't go through them all, but it's really how do we make, in fact, data when we're sharing it FAIR compliant. And it is things like being able to do microattribution for individual submissions at any level of granularity, being able to track every data item separately inside the Genomic Data Commons, and be able to track how that's been used. And I'll come back to that point in just a second. I also think it's really important, and these are by no means all the people involved. These are only the leads. At the University of Chicago, there's been more than 45 people involved. I'm not sure how many at Ontario, but it's been dozens. Likewise at NCI, there's been a tremendous number of people involved in this. So it does take teams. It is team science. And again, it's really important to recognize how many people it takes to do so many things today. Something else with the Genomic Data Commons, when it was announced, we've had now a number of different groups want to release their data through the Genomic Data Commons for public access. And the one that in some ways is the most interesting has been Foundation Medicine, a for-profit company, has agreed to release about 18,000 genomic profiles for public access. Likewise, Multiple Myeloma Foundation has released the COMPASS study, which is a far more detailed and longitudinal study of multiple myeloma patients, all through the Genomic Data Commons. And AACR Genie has released almost 19,000 genomic records through the GDC. So we're seeing a lot of different groups release their data through the Genomic Data Commons, and we think that's really important. However, if we really want to support FAIR, we really want to be able to track from not just data deposition, but through data usage and how people can make use of it, and also facilitate that use. And that's really been the idea of the NCI Cancer Genomic Cloud pilots. So there are three different pilots I'll go into, but it's about both making the data available in a cloud environment, letting people do compute at the data so you don't have to download 1.5 petabytes of data, and making sure that all that happens in a secure and well-tracked system. And it really is about democratizing access to all those data. So the three pilots are Gadi Getz at the Broad Institute, Ilya Shmulevitz at the Institute for Systems Biology, and Dennis Corral at Seven Bridges Genomics. And they each use a little different infrastructure as well as different commercial cloud providers. So the Broad and the Institute for Systems Biology use Google, and Seven Bridges uses Amazon. And again, I think the focus there isn't so much on the cloud providers as it is being able to get this securely and successfully deployed in different cloud infrastructures. And how do we make sure that, in fact, we as computational scientists can access and utilize those infrastructures appropriately. And to make some of that happen, we think it's also really important you can bring your own data and co-analyze those data with the publicly available data. And what we'd like to do, and we're still figuring out how to make this happen from a business model perspective, is that if you're willing to make your data openly available, it's easier and cheaper for you to do that than if you want to keep your data proprietary. So again, stay tuned as we figure out how to really make that happen. And again, there are a lot more people involved than the few people I've shown here in each of the cloud pilots, and even just managing the cloud pilots at NCI. So I want to thank all those people and many more behind them. So the Genomic Data Commons is really just the beginning of this, and Tony's going to talk a bit more about the Research Data Commons this afternoon. But it's really about taking a lot more data than just the genomic and clinical data and make it accessible and make it interoperable in very different ways than we've ever done before. And of course, we want to make sure that it's not just about computation. We want to make sure it's accessible to biologists, to clinicians, to whoever wants to be able to use infrastructure like this. And that it's easy to be able to cross-analyze the various types of data and various data sets that go into this kind of an infrastructure. So we want to make sure that, in fact, as data is ingested into these systems, that they're interoperable with each other and they've been well annotated, and that annotation is as automated as we can possibly make it. Because again, something that becomes a big scaling problem for all of us is when human beings have to do too much work as data goes in. And right now, with the Genomic Data Commons itself, we're still learning what some of those rules are, and it's a little more tedious and manual than I would like. The other point, and I won't dwell too much on this, is that we want to make sure that there isn't just a single set of nodes, that it's easy for people to stand up new data commons nodes. And again, I think this is going to be a really important part of the NIH data commons as well. I had the privilege to sit in a meeting with the various folks at NIH that have been planning the NIH data commons, and there was a comment made that we can scale from 3 data sets to 30, but we can't think about going past 30. And I think that the reality is we need to go to 300, to 3,000, to 30,000, to really every single person that generates data anywhere in the community, and give them a place to have that be interoperable and shareable. So I think there's a big scaling problem that we're still grappling with. I mentioned the SEER registry. This is just a little graphic, and I'm afraid it has too many automations here, which I always forget to work. But the idea is for the SEER registry that we have access to population-based data that's demographics on patients that have cancer, pathology, there's molecular characterization data, there's progression, recurrence, survival, death. And all that data can really be very useful in understanding cancer as a disease and the impact of changes in therapy. And again, I think that's the whole focus that I've been hearing more and more about in the various scientific literature is a focus on real-world evidence. So it's how do we make that happen? How do we make that easy to do? So again, I want to just say that genomics is the beginning of precision medicine. It's clearly not the end. One more point I want to bring up is that the scale of science that we can do has dramatically changed in a short period of time. And I think, again, we all know this, but it's easy to forget it because it's happening so fast. But back in 2001, to sequence a single human being, or actually a collection of people, but to end up with one single reference genome was about $3 billion US. And in 2010, the NIH announced a 1,000 Genomes project for way less than $100 million. So that was a dramatic decrease in cost and an increase in the scale in which we can generate data. And then 2 years ago, the NIH again announced what's now called All of Us, Precision Medicine Cohort, and looking at 1 million people and thinking about being able to sequence 1 million people. At the same time, the VA, the Veterans Administration in the United States, announced a 1 million vet program. Genomics England announced a 100,000 genome project. it's clear that we're doing things at a very different scale in a very short period of time. And I think it's pretty obvious to many of us that this doesn't stop until we start sequencing every person, and maybe we start sequencing people every time they come into the clinic, because it becomes cheap enough that that's maybe the cheapest diagnostic we can do. At the same time, it's also clear that we're not data sharing the same way that we could be, or at least as effectively as we could be. So this is a graphic from the Harvard Business Review, which I don't often get a chance to give graphics from the Harvard Business Review. But this is a really interesting that this is public data, this is more private data, and you can see the different data sets, and this was done now a year ago. Different data sets. Most of the data sets that are public have very little longitudinal data and relatively little clinical data. One of the few exceptions is the MMRF COMPASS study that I already mentioned. And we really need to get more and more clinical data up into this area of the graph. So we need to have a lot more complex phenotype, complex clinical data, and done over time. And again, that's been one of the focuses for all of us, the NIH Precision Medicine cohort. So I don't think I need to emphasize this too much more or convince you of this, but really biology and medicine are data-intensive enterprises. I think we all recognize that. The scale's rapidly changing what we can do. And in fact, data and computing and information technology are pervasive. They're pervasive in the lab, they're pervasive in the clinic, they're pervasive in our homes, and they're really pervasive across the entire society now. And that's been, again, a remarkable change. I was at a patient advocacy meeting 2 months ago, and what's been interesting is hearing the folks talk about health disparities and saying, you know, access to data really can decrease disparities. 5 years ago, that was not the case because it seemed like not everyone had equal access to technology, but that's changed, again, so incredibly rapidly. So I'm going to really quickly go through perhaps what's going to be an intro to the next session. I hope it will be. That's what I intended it to be. And that's give you a little sense of why I think machine learning is so incredibly important in computing. So as we've been generating more and more data, as we've been making more and more complicated decisions in the clinic, what is perhaps not obvious from this slide is human cognitive capacity hasn't changed in the last 30 years dramatically. We change on evolutionary timescales, not in weeks or months or decades. And at the same time, we're asking now clinicians to make so many more decisions based on so much more data. And that's only going to get more and more difficult to manage. So how do we help clinicians manage that? So this is a publication in Nature the beginning of this year by Sebastian Thrun and colleagues. And it was really looking at machine learning of dermatology images, trying to identify melanoma. And the outcome of that training was that the deep learning network that he put together was as effective as the best dermatologist. So I think what's interesting about that is it says we don't need to ask dermatologists to look and grade images. We can do that computationally, and we can do that as effectively as anyone in the clinic can do it. So we remove a very complicated process from the dermatologist and give them a yes-no answer, and that's much easier to make a decision on. At the same time, there's been some very interesting, I'll call it hyperbole, in the public press about AI. This one was entitled AI versus MD. This is Siddharthi Mukherjee, who wrote The Gene most recently, and also The Emperor of All Maladies a few years ago. And although that's a very provocative title, In fact, he points out that this is inevitable and it's not a bad thing, that we can start to use AI or machine learning to really extend what people do. He also made the point that this has been known for a long time. Back in 1945, the British philosopher Gilbert Ryle identified or classified two kinds of knowledge, what I would call expert systems, or knowing something, versus experiential learning, which is knowing how to do things. And he gave a nice example of learning to ride a bike is very different than understanding the physics of how a bike operates. And I think it's also important to realize that human cognition, at least in my view, there are at least 3 levels of, it's not just learning, the rule-based learning or experiential knowledge, it's also us asking why. And that right now is outside the reach of even the best AI. So from a machine learning standpoint, I hope you agree that if you like C3PO, he was really simply a decision aid for human beings. If you don't like C3PO, perhaps you like R2D2. Again, a decision aid for people as they were going through a complicated set of processes. So again, I think it's important to realize that AI is actually here with us already. Our phones actually help us get around Prague if we've never been here. And we just expect that now. And I don't think that's bad. I do want to mention one last thing. This is Apollo. It is a proteogenomics cancer project where we're learning from both proteomics and from genomics when patients present with different cancers, what are they really presenting with, and how can we optimally treat them? What's been really exciting is this is again part of the Cancer Moonshot, and we've had now 11 different countries sign 10 different MOUs with us, with the NCI, and there's a data sharing pledge that every country takes that signs these MOUs to share the data openly across the world. So again, we think this is going to really change our understanding of proteomics and genomics. And I see I have run completely out of time, but I will leave you with the thought that really what we're trying to do is create meaningful cancer computational predictive models, things that we can deploy widely and that will really help us make better decisions in the clinic. And with that, I will happily take questions if we still have time.